I am so deeply honored to be presenting on this subject, coming to you from the recorded past and the other side of the international dateline in real time. We're also a day into our respective equinoxes, vernal for the morning southerners, autumnal for the nocturnal northerners. I have no relevant relationships and I won't be naming any medications on or off label. To be honest, I found chemotherapy to be a lot less fraught than chemotherapy quite early into my training. I was especially moved by the discipline that was channeling the renewable and altruistic resource of blood from one person into another in a safe and personalized way, and then enthralled to learn the deeper significance of the compatibility systems themselves. So though my practice and research focus mostly nowadays on hemovigilance and immunohematology, my journey as a fellow and junior staff some years ago chased the relationships between human blood phenotypes and outcomes in Plasmodium falciparum malaria, a formidable gene shift force as an infectious disease that is so predatory in the immunonaive young. This wouldn't have happened without the power and devotion of my mentor, Sunny Zeke, and our on-the-ground super stakeholder family in the sub-Saharan nation of Uganda. We're most indebted to the phenomenal Doctors Musoka and Debanji, and to the funders of the cytoadherence in pediatric malaria study. In 2010, I took my first trip to Australia to share the results of the CPM study at a conference in Melbourne. This was such an earnest pilgrimage that my spouse and I got certified as divers in the cold monochrome of Ontario waters. And in the privilege and wonder of beholding the Great Barrier Reef directly after the conference, we were changed forever. It was truly altered states to see this pageant of life and to rejoin that water-breathing fork of evolution that was out of sight and out of mind until then. Australia intensified my appreciation for the evolutionary principles of selection, adaptation, imitation, form, and function, which, as we'll see, are so prominent on the red cell. So let's dive in on a plan to survey the red cell, malaria, and the star systems of ABO, Wiener, Duffy, a smaller miscellany, with parting thoughts on the past and future. So though it may seem distasteful to joke that the largest organ of the human body is its acquired resident microbiome, equal in weight to an adult hamster, the most abundant cell type made by a healthy human is their red cell mass. To be ultra reductionist, we have two halves, the HLA side and the RBC side, with the latter devoid of invader antigen presentation capacity, mitochondria, and thus oxygen-using Krebs cycle, and a nucleus. This dualism is useful if you are considering the hazards of viable lymphocytes in transfusions or transplants, a T-cell mediated graft versus host disease, or plasma cell mediated hemolytic passenger lymphocyte syndromes targeting red cells, respectively. I like to call the red cell our metabolic saint and magician, because at the right hematocrit, it alters an equal volume of water to carry as much oxygen as air itself. And not only are these red discoid boomerangs delivering oxygen, they're carrying back carbon dioxide to the lungs, making them little micro lungs, while also having talents in nitric oxide co-production and shipment. To live as long as it does and to be able to squeeze through capillaries narrower than their own diameter, and especially to endure the editorial fenestrations of the spleen, these have got to be both strong and flexible, i.e. tensegrity structures. I don't want to sound too parentalistic here, but do you appreciate your red cells? The last time I went to a museum before the pandemic was at Toronto's ROM, which was showcasing the science and mythology of blood and bloodsuckers. There, I learned about the Aborigines legend of an exsanguinator monster called the Yaramayahu. It turns out that the majority of animal phyla have independently and convergently evolved blood-feeding biologies. So the Aborigines were onto something. Weirder still, in the protist kingdoms, the phyla there have also evolved convergently toward parasitism, with some of the apicomplexans, Plasmodia or Babesia, hitching a ride in blood-feeding arthropods, like vampires within vampires. Nature informs us again and again that parasitism is a breakthrough, so the evolutionary arms race in counter-adaptation must proceed. Can you blame a bug for picking the red cell as its cell of choice? Are we to be surprised at how red cell features align with selection pressures? Plasmodia have been assailing their corresponding land vertebrates of choice for millions of years after their co-evolutionary Big Bang dance. For the 108 billion humans who've existed since 50,000 BCE, mosquitoes, the carrier of Plasmodia, have been estimated to kill half of all who've ever existed. 1 in 7 humans are now imprinted in one way or another with a malaria adaptation, most affecting the red cell in either a single or double dose requisite advantage load. Taken together, a quarter of the total variability in malaria severity is attributed to host genetics. The WHO defines severe malaria as any one of these features, where of course, the more that are present, the higher the case fatality rate, which ranges from 10 to 20% with severe features. 1 to 2% of African children will develop severe malaria with a 15% lifetime risk of requiring a hospital admission for their infection. Malaria starts at the skin and seeds to expand in the liver. But where it gets you sick and into astronomical copy numbers is in the erythrocytic phase. The great virulence leap of falciparum compared to the other species that infect humans is the capacity of this one to invade all red cells and to tack infected red cells down to avoid flow into the spleen. Adhering to the endothelium and to a shroud of uninfected red cells will bring pathologic vaso-occlusion and enhance cell-to-cell transmission. So to study if a host adaptation helps you, you have to ask if its anti-invasion power is operative, or if it's a trick to limit red cell effects once you've been infected, because you might otherwise miss your effect discovery, so to speak. Young humans who are seeing malaria for the first time and people who are asplenic are so prone to getting sick because the infection takes off faster than the innate immune system can act in the period before the adaptive immune response gets developed. By this, I mean the monocytes and macrophages especially. Remember, these are densely situated in the all-important editorial spleen. So this is where the simplest red cell stands, with the interior containing hemoglobin and enzymes and an exterior built of membranoskeleton and studding antigens. These have adapted either to toggle down the fitness just enough to keep functional when uninfected, but to look really unfit all the sooner when infected, so as to maximize the custodial power of the spleen and monomax. The world of binding adaptations, in turn, will influence portals of entry or evade the parasite-encoded sticky points on infected red cells as they interact with the host. Let's connect ABO to malaria in the punchline up front. Here is PFEMP, the promiscuous Velcro-like knob that erupts on a falciparum hijacked red cell. You can see adherence, clumping, and rosetting. Here are the host latch points. On red cells, the ABO system at 5 million copies per cell presents the most abundant and seemingly stickiest of all targets within the A antigen in particular. In the CPM study, we took an integrated look at the host ligands in blue. Early in our hominid evolution, wild type A mutated out to B, then O, with a differential ascendance of the latter and scores of weaker, less branched versions of A among those with conserved A type, as if to intimate a running away from anus. Taking Haldane's eyes, the ratio of O over A was exceeding 1, where the tropical infection remained intensely endemic, with a preservation of the Exodus A majority that migrated to climes subjected to less of this selection pressure. O is a big leap, and you need to be homozygous to see its advantage in the reverse type. Nevertheless, it's reasonable to hypothesize that you're armed with parasite-neutralizing isoagglutinins against A-like structures. However, the epidemiology does not support an ABO effect at the level of blocking infection. Instead, there is good clinical evidence on mitigating severity with molecularly compelling effects in vascular biology models. We hypothesized that O's are untethered, and well-secreting non-O's can mitigate the exploitation of A or B binding. Though severity is not equivalent to death, and death is what shifts gene ratios, we reasoned it's a decent proxy. And in our pre-study systematic review, we saw the mirror O advantage, A disadvantage effect. In the CPM study, we saw an ABO gradient effect on severity, an antigen dosage effect with less of the cytoadhesive syndromes, that is cerebral malaria or systemic lactic acidosis respiratory distress, in the 10% of A's who were weaker subgroups of A greater harm from having an A allele than the benefit from being sickle trait positive, a statistically stunning difference in the ABO distribution of healthy adult donors versus the pediatric cohort experiencing malaria, and finally, the mortality effect, with 50% of decedents having an A allele versus the less than 30% of uncomplicated malarias. Said another way, the odds ratio for death versus uncomplicated malaria was 2.3 in A versus O. ABO zygosity effects are less apparent on the red cell. So how the O mutation in the codominant ABO system got to enough homozygous people to experience the survival benefit isn't clear, unless one takes the platelets as an important part of the malaria glue and hypothetical pathology escape by expression dosage, which has been described by us and others, but not yet proven in malaria outcome mapping. Whereas medications all too often flow from prohibitively expensive development and purchasing pathways, with parasites invariably developing resistance by their operating scale, I'd like to point out that there is no drug to prune off PFIMP once it's formed. ABO chemotherapy, on the other hand, presents a choice of O red cells instead of type-specific blood and soluble A and or B containing plasmas as an anti-adhesive dust. This would be a compatible way of imitating what evolution has taught us and work as a bridge until parasiticide is complete. Tempering the majesty of onus in malaria, Stowell and Stowell published beautiful reviews on Flipside infectious prices, showing the vulnerability O. Lewis B positive secretors experience for H. pylori, though that is an infection that will not cut you out of the reproductive gene pool. Granted, the vulnerability of O non-secretors to cholera, an infection that does still kill about 100,000 people every year, which is about a fifth of the world's annual malaria fatality count. They also pointed out, though, that it's not a total loss if you lack isoagglutinins, as galectins back you up with neutralizing germ power too. ABO has an important relationship with hemostasis and thrombosis. Von Willebrand factor is a pivotal molecule that gets shellacked with one's ABO type, with A or B sites rendering it tougher or slower to proteolyze. Almost a third of the variability in von Willebrand factor and factor VIII levels is dictated by the ABO type, with the Gaussian distribution of OVWF levels running near or into type 1 von Willebrand disease levels, and the non-Os conversely exhibiting higher thrombotic event rates in scores of papers. The ABO type seems even to influence how quickly platelets roll to an adherent stop on the endothelium. No talk is complete without mentioning COVID, and ABO pops in here. One's ABO type ends up on the viruses they make, so that group O people have virus neutralizing isoagglutinins, whereas non-Os might be infecting with a comparable viral ABO that enables entry. As mentioned, non-Os have more thrombotic firepower. Couple that with the inherently thromboinflammatory nature of COVID, and you may be seeing additive effects. 7 years ago, the great Fumijiro Yamamoto, internationally renowned for his elucidation of the molecular genetics of the ABO system in 1990, published this paper on ABO and related gene evolution across all life forms. They did homologous gene sequencing with comparative chromosome structural analyses, phenomics, and substitution mutagenesis to correlate codons specific for the immunodominant sugars. In this phylogenetic circle, colored lines show the 5 glycosyltransferase fan-outs, with the dots representing the 4 non-mammal orders. You can see that mammals have the full deck of enzymes, and that only mammals, amphibians, and reptiles have ABO systems in phylum Chordata. They deduced FUT2 to be the most primordial, with ABO arising after the fish-amphibian separation, with a shift from multigenic polymorphism, in variable AB representation that is, to unigenic constraint and thus allelic polymorphism in primates. No surprise, there are prokaryotes taking ABO sides, many being members of our microbiome, thought but not unambiguously proven to be the stimulus for the development of our reciprocal isoagglutinins. Not all bacteria express ABO though, a gene which entered by horizontal and vertical transmission events with conservation because of the advantage of stealth by mimicry. The counter-evolutionary drop-off of A and B suggests neutralization power for us hosts in some regards. The largest study investigating ABO and RHD type with human disease occurrence was conducted by Gustav Edgren and e-published recently. Though limited by being a Nordic non-malaria and pre-pandemic data set, 49 diseases popped out with ABO and 1 with RHD. The significance volcano plots on condition-specific incidence rate ratios showed clots with A and AB, pancreatic cancer with A new finding of kidney stone protection with B, and pregnancy-induced hypertension protection with O and D negativity. There's still more to discover on ABO though. Will we have ABO-friendlier platelets selected for lower by single dose A or B expression and less isoagglutinin content by plasma volume reduction or platelet additive solutions? Will we figure out how to mitigate the risk of high dose IVIG hemolysis in non-O recipients by altering the product or how we give it? Will we standardize ABO crossings in solid organ transplants? Will we have better and harmonized truth tables for hybrid ABO types following mismatched BMTs in their various phases, with rules for multi-type crossings in serial BMTs or mixed solid organ transplant BMT chronologies? Will we deploy enzymatically converted to O products for inventory and allocation universalism? Let's flip to Wiener now, a heterotrimer complex of CE paired with its tandem D gene product if present, or 2 copies of itself otherwise, coupled with 2 RAG glycoproteins for about 30,000 of these complexes per red cell. The CED sequence sits at chromosome 1p, transcribing a 10 exon sequence for translation into a 416 amino acid 12 transmembrane pass protein with near identical sequences and a lesser degree of similarity with RAG from chromosome 6. What is this complex supposed to do? The phylogenetics suggest ammonium transport, though Westhoff showed a human-specific difference in the critical motif for that function. Because of how enmeshed RH is with other structures doing things like carbonate transport or pinning into the cytoskeleton, it's hard to know if this is an evolutionary appendix of sorts with over-ascribed meaning by its structural associations. When I teach learners about the RHCE protein and the C and E types, I underscore that these are parts of a parentally inherited structure, akin to small or large, symmetric or asymmetric ears on Mickey Mouse. For the D pauses, you'll have the foundational R0 of Africa, Eurasia's R1, Beringia's R2 with the rarer RZ, their CE configurational antigenicities, and then the extremely rare D-, lacking any version of a partner CE. CE without D is more common, a deletional event tracing to Basque country, with the zygosity deducible in D pauses by detection of the up-down hybrid rhesus box in the stereotypic European allele. In addition to this mode of D negativity, there are two dominant African modes by a 37 base pair duplication in D psi and a DCED hybrid. D negativity is rare in Asia, with some apparent D negs being misrepresentatively so and actually constituting the del state. The good news is the reliability of big C and big E negativity in D negativity, but the bad news is the challenge of alternative D neg zygosities when they alloimmunize and need blood. Do you know your full wiener type? As a diploid creature and daughter of an A-pos dad and an O-neg mom, I'm a group A R2r. My blood would not be the best choice for the former US president. Let's switch from the D-neg white male to the black female, more often D-pos, but not straightforwardly so. European RH variation is a comparative monotony when looking at the diversity of wiener variation, be it altered with unknown significance, a sensitization vulnerable partial format, or non-expression. This diversification is founded on an ancient duplication event resulting in tail-to-tail orientation conducive to gene conversion with hybrid allele formation at recombination opportunities. Despite this, SNPs still outnumber the hybrid variants, suggesting then the imprint of great lengths of time. The further away you get from Africa, the less diversity there is, speaking to narrower splays of founder groups. This tracks us back to Mother Africa as the seat of our origin, a potently anti-racist position in evolutionary science against the notion of a white Adam and Eve. An important concept we bring to the table is the quantitative spectrum of reduced or weak expression and the qualitative spectrum of exofacially different antigens compared with a wild type, and then the nuance of overlap. Many variations in D and CE have been cataloged with molecular or new antigen names that drive some or maybe most of our peers crazy. What matters for recipients is not so much the presence of a variant, but the load, with homozygosity or compound heterozygosity being the problem. We'd love the shelter of a wild type haplotype, without which we might end up with a partial little E state. The names of the variant RHCE genes that might look little E-pos serologically, but be capable of generating anti-little E-like antibodies are shown here with Bantu Shabalala and South African Bastien. It was stirring to see this recently e-published paper on wiener paleogenetics in our ancient hominins. In Western Vindhya Neanderthals, Eastern Chagirskaya and Altai Neanderthals, and in Denisovans, partial D and RHCE were seen, suggesting an African offshoot with a hypothesized time bomb for HDFN-driven extinction. Interesting idea. Let's move over to Duffy, recently renamed ACKR1, a differentially expressed chemokine receptor. Haldane's eyes on the distribution of the GATA promoter mutation upstream of the dominant Duffy B form of the gene in Africa shows a remarkable spread of homozygosity, which leads to a vivax and Knowles-eye uninvadable erythron by lacking the parasite's typical portal of entry. Though vivax and Knowles-eye are not as deadly as falciparum, these data suggest that perhaps these species were once more virulent to have compelled so much change. The effect of the genotype goes beyond red cells not expressing the chemokine receptor. This appears to explain so-called benign ethnic neutropenia, the mechanism of which was worked out 3 years ago. But ethnic to whom? Otherizing and racializing are impositions whose roots we have to question. The goal of science should be the pruning of inequities, not the exacerbation of them. The undercurrent coming through all of this antigen talk is a call to personalization rather than pathologization. After all, we vary more person to person than from race to race. I know there are hot politics on the study of health matters associating with geographic origins and current environmental contexts. The voices on how to respectfully speak about and collaboratively engage in questions and answers are rising for all of us to listen to. We can't deny what we see about the Duffy-Vivax connection, and we also can't look away at what the possible prices are to natural or induced gene changes. Maybe benign is the wrong word. If the Vivax triumph carries a theoretical falciparum price, or the physiologic risks of an altered chemokine environment. These questions are not to silence, but to compensate and solve certain double-edged swords. So is there any falciparum specific invasion resistance phenotype? It turns out the answer is yes, but not at the spread of Duffy's altered GATA. Here's a snapshot of the Democratic Republic of Congo and the DAN2 variants across Middle Africa. Glycophorin variation is not only seen in continental Africa, but in Asia, where malaria is also a problem. And there are at least 50 antigens in the M&S system today. So to summarize our most variable systems, Wiener and glycophorins have explanations now for how and why their inherent variabilities are set up. For completeness, I'm flashing you with the Diego Band 3 anion exchanger 1 that also plays a portal resistance role in certain alterations. Another interesting evolutionary twist is the loss of CR1 or CD35 in the Knops-Helgeson type. By missing this ligand, there is less cytoadhesion, but more hemolysis. Syndromically, this would seem a better choice, and indeed, we see this polarization in regions with higher versus lower transmission. The last thing I'm going to mention is KLF1, which coordinates the expression of many red cell structures, a go-to route if you are looking for inhibited or suppressed antigens to serve certain L-immunized patients. So here we are in 2021, staring down the barrel of more and more types and antibodies to know about. Every year we realize that the screen on our Griffin screen is tasked with recognizing more and more of these variations. And it's not just, are you positive or negative? It's if you express or tolerate more than you would seem to, or express differently and misleadingly with a vulnerability. The geno-pheno correlation game is not a petty exercise. In my practice, I'm humbled at the frequency and severity of red cell rejection events in our patients with sickle cell disease. There are unfair disadvantages at so many levels. How can you not work toward offsetting them in medical modes of affirmative action? We need better representation, as shown in the bloat of white DNA in genomics research. At the same time, representation must not be exploitive, but reparative and engaging. The most numerous, biomechanically resilient, service-oriented frills-stripped cells of our body carry the antigens of interest to blood bankers. Wild type A and ABO is being weeded out or weakening. Structural and genomic accidents in time have driven Wiener diversity. Reduced parasite invasion, reduced parasite adhesion are founded on certain blood systems. Changes come with costs. Struggles past impress, form, and function upon us and our kin. Look upon differences as a call to compassion and understanding. And with that, we will surface. And I thank you so much for diving with me.